Hello students, welcome to EPG Pathshala. I am Dr. Savita Yadav from Department of Biophysics, AIMS. Today I am going to talk on module Gel Filtration, Principle, Methodology and Application from paper Techniques in Molecular Biophysics 1. So what are we going to learn from this module? Theory of gel filtration chromatography, how to select a media, how to pack a column, and what is the procedure of gel filtration chromatography, and which are the factors which affect resolution of gel filtration chromatography, and what are the applications. Gel filtration chromatography is used to separate proteins and peptides and oligonucleotides on the basis of their size only. It is also known as size exclusion chromatography. In gel filtration, as sample moves through the bed of porous beads, molecules of different size diffuse into beads to a greater or lesser extent. Small molecules enter further into the beads and thus elute slowly. In comparison to that, large molecules enter less or move directly through the void volume and thus elute fast. Gel filtration chromatography is widely applied for molecular size analysis, for separation of components of a mixture, as well as desalting and buffer exchange. There are numerous factors like column dimension, media, packing density, particle size, distribution, buffer, sample preparation, flow rate, and fraction size that affect the separation quality in gel filtration. Successful separation of analyte highly depend on selection of appropriate media, sample concentration, volume, tube length, flow rate, and fraction size. What is the history of gel filtration chromatography? Nearly 50 years after Sweat discovered column liquid chromatography, gel filtration chromatography was first recognized accidentally as a new method of molecular separation. In early 1950s, some researchers noted that neutral small molecules and oligomers were eluted on the basis of decreasing molecular weight from the column of cross-linked polystyrene ion exchange resins. Duell and Lindquist also reported similar results for the elution of low molecular weight molecules using cross-linked galactomannan and starch granules. Then in 1955-56, Lath and Rathwan used columns packed with starch granules to study the elution profiles of low and high molecular weight polysaccharides and proteins. And surprisingly, they observed that high molecular weight molecules eluted faster than low molecular weight molecules. For this work, they were given credit of performing the first biopolymer size separation. Lath and Ruthven called the process gel filtration. In 1959, two biochemists, Perflodin and Jerker Porath, synthesized a series of cross-linked dextran resins with different pore sizes for size separation of peptide and oligosaccharides. The polymer scientists in early 1960s used hydrophobic packing for gel filtration of synthetic polymers. It was referred to as gel permeation chromatography during that period. Molecules was first performed by a brewer who used swollen rubber granules for the separation of hydrocarbons. What is the principle of gel filtration chromatography? The separation of molecules on the basis of their molecular size and shape is achieved by gel filtration chromatography. It uses molecular sieve properties of various porous resins. Large molecules that are completely excluded from the pores pass through the void volume. Interstitial spaces between the resin particles and thus they elute first. While smaller molecules get distributed between the mobile phase inside and outside the beads and therefore pass through the column at slower rate. Thus they elute in the last. Trapping of the mobile phase inside the bead solely depend on porosity of the resin beads and size of the molecules to be separated. Thus, The distribution of a molecule in gel filtration column of cross-linked beads is determined by the total volume of mobile phase both inside and outside the beads. In case of gel filtration chromatography, the distribution coefficient of a molecule between the inner and outer mobile phase is the function of its molecular size. If a molecule is sufficiently large to completely exclude from the mobile phase between the beads, then distribution coefficient K will be equal to 0. On the other hand, if molecule is so small that it accesses to the innermost mobile phase deep in the beads, then distribution coefficient K will be equal to 1. For others, the value of K will vary between 0 and 1. This variation of K between 0 and 1 makes it possible to separate various molecules present in a mixture within a narrow molecular size range. Fractionation range, operating pressure and temperature, flow rate, sample viscosity, pH range, and tolerance for detergent and salt, etc. are important for selecting gel filtration media. The column or media should be able to withstand the pressure and temperature at which we want to separate a molecule. It should also be able to withstand flow rate. Suppose one has taken high flow rate, it should not be like that column get broke because of high flow rate. Since it is a gel filtration chromatography, we are concentrating the sample, and after concentrating, the sample has become slightly viscous. Therefore, it should be able to pass through the pores of the gel, and gel filtration media should be stable in wide pH range, say from 3 to 10. And it should be tolerant to salt as well as detergent, because in gel filtration chromatography, sometimes salts or detergents are added to separate subunits. So these are important consideration one should have for selecting gel filtration media. The commonly used media for gel filtration chromatography are dextran, polyacrylamide, and dextran polyacrylamide, and agarose, and they are available for large range of pore size for separation of macromolecules of different sizes. The table shows different commercially available media for gel filtration chromatography. Dextran based media is available in the market as Cefadex. It is available in different range starting from G50 to G200, and molecular weight range is from 1.5 to 600 kilodalton. Polyacrylamide based gel media is also available in the market in the name of Biogel P. It is available from the size P10 to P200, and it can also have molecular range from 1.5 to 200 kilodalton. Dextran and polyacrylamide gel media is also available commercially, and they are known as Cefacryl, and they are also available in different sizes. Another agarose based media is also available commercially. It is known as Cefrose or Biogel A, and their range is also from 10 kilodalton to 5000 kilodalton. The table shows again different commercially available media for gel filtration chromatography. A gel with a smaller range of pore size gives higher resolution, while a gel with a wider range gives lower resolution. However, the benefit of using wide range gels in fractionation of higher range of sizes when molecular weight of analyte to be separated are unknown. Packing of the column can be divided into three parts: swelling of gel, pouring of gel into the column, and equilibration of the column. Column dimensions are very important. Resolution increases with the column length. So in gel filtration chromatography, the column length should be large. Now, how gel should be swelled for gel filtration chromatography? Add appropriate amount of filtered double distilled water to the gel matrix depending on the swelling capacity of the beads as per manufacturer's recommendations. Once the gel has swollen and settled down in the bottom of the beaker, decant the double distilled water on the top. Then add appropriate buffer to the gel. You have to mix it gently. You do not have to use magnetic stirrer for mixing, because if you use magnetic stirrer, it will break the beads and then separation will be poor. Now wait for it to settle down. Once it is settled down, you decant and resuspend the gel in equal volume of the buffer which you are going to use. Now degas the gel in order to remove trapped air. If you want to do rapid swelling of the gel, it can be achieved by heating the slurry at 90 degrees centigrade for 5 hours. Swelling at room temperature is considerably slow and takes approximately 72 hours. All the buffers and gels has to be degassed to avoid air bubble formation, since it will cause uneven packing of the column and it will give rise to poor resolution. All the buffers has to be filtered before using it in gel filtration chromatography. We have already learned that how to swell a gel for gel filtration chromatography for packing of a column. Now next step is pouring of the gel into the column. Mount the glass column vertically on a metal stand and adjust the bottom adapter. Once you have mounted it vertically straight on the metal stand and adjusted the bottom adapter, add some buffer to the empty column. What will happen when you add buffer to the empty column? And if it is passed through the outlet tubing, your trapped air bubble in the path will be removed. Now block the outlet of the tubing and pour the gel slurry to fill the column up to the required height with the help of glass rod. The glass rod should be used in such a way that it touches inner wall of the column. This avoids introduction of any air bubble. A gel reservoir should also be used to smooth and continuous pouring of the gel. Allow it to settle down, decant the excessive buffer and the gel and put the top adapter on the top of the gel. Once now column is packed, now you have to equilibrate the column. Attach the top tubing to the peristaltic pump and buffer reservoir and keep the bottom tubing to the waste. The operating pressure should be according to the manufacturer's recommendation for the gel matrix. It is always better to keep flow rate slower. Choose the buffer as per your purification needs and equilibrate the column with 2-3 bed volumes of this buffer. The total column volume Vt is referred as bed volume, while the space between the gel particles is called void volume V0. In a well-packed column, the void volume is about 1/3 of the bed volume. The large molecules directly elute through the void volume, while the small molecules, which gain full access to the complete mobile phase, will elute after the one bed volume and they do not separate. The column volume, total column volume, if you subtract void volume from the total column volume, what you will get is volume occupied by the gel, including gel matrix V gel. So stationary phase Vs can be written as Vt minus V0 minus V gel. This is the journal setup of gel filtration chromatography, in which you can see the peristaltic pump is attached to buffer reservoir on the one side and it is attached to gel filtration column on the other side. Peristaltic pump is important for controlling the flow rate. Gel filtration column at the bottom through tubing is attached to fraction collector with detector, so that whatever peaks are eluted out are detected by the detector and then elution profile can be obtained. This is the manual setup for gel filtration chromatography. Another picture is showing the automated setup for gel filtration chromatography, where one can use directly sample injector, pre-packed column and a screen on which results are displayed. Let us discuss process of gel filtration. The first step is buffer selection. Any buffer in which analyte to be separated is stable can be used as pH, ionic strength and composition do not significantly affect resolution, though the buffer should maintain the buffer capacity and constant pH. Low strength salt can be added to the buffer to avoid any interaction with the gel particle, or if there is an ionic interaction between molecules to be separated. Sample buffer does not have to be same as buffer in the column. Extreme pH conditions and ionic strength should be avoided because it may affect the gel as well as separation. Prior to using, all the buffer should be filtered through 0.22 millimicron membrane, and some commonly used buffer in gel filtration chromatography are tris-HCl and phosphate. Next step is sample preparation and loading. The sample should not be diluted, it may cause band broadening. Therefore, it should be concentrated. However, it should not be that concentrated that it precipitates. Ideally, 1% of bed volume of sample should be loaded onto the column. The sample should be clear, free of any debris, and it should be filtered prior to loading. To load the sample manually, you have to remove upper adapter and remove the excess of the buffer on the top of the gel without disturbing the gel surface, and then gently pipette in the sufficient amount of sample and allow the sample to pass through the surface. Once the sample has passed through the surface, you have to see that the surface of the gel does not dry. Then you have to add little buffer on the top of the column and then set the upper adapter. The above process of loading sample onto the column can also be done by automated sample injector. If you use sample injector, your surface will not be disturbed and your resolution will be higher. Once you have loaded the sample, you have run gel filtration, elution has to be done of the same sample. So for this, a fraction collector is attached to the system to collect the fractions of the elution. The sample is eluted isochronically using a single buffer system. The length of tubing connected to the fraction collector should be small, and small size of sample fraction size should be collected. It should be 1 ml or less than 1 ml to avoid mixing of peaks. The detector connected to the fraction collector analyzes the separation, and the total elution volume should be equal to or more than 1 bed volume. Elution volume VE is volume of the buffer that elutes from the column before a particular peak in the elution profile appears. The distribution coefficient Kd of an analyte in a gel filtration chromatography can be mathematically expressed as Kd is equal to VE minus V0 divided by Vt minus V0. VE is elution volume, V0 is void volume, and Vt is total column volume. After completion of each run, the column should be washed thoroughly to remove any analytes left in the gel. Ideally, it should be done by 1 to 2 bed volumes of buffer. For long-term storage, antimicrobial agent should be added to the buffer. How do you calibrate a column in gel filtration chromatography? It is important to calibrate a column for determining exact molecular weight of substances to be separated. For accurate determination of molecular weight of molecules to be separated, gel filtration column can be calibrated with standards of known molecular weight. For this purpose, a calibration curve is drawn by plotting log of the molecular weight of standard proteins against their respective ratios of elution volume to the column void volume. The calibration standard should be selected in such a way that there is same relationship between molecular weight of standards and molecular size of the substance of interest. There are numerous factors which affect separation quality in gel filtration. An important factor I am going to discuss here is first is column dimension. The column dimensions are very important for better purification. For gel filtration chromatography, resolution increases with the column length, while the column diameter increases the capacity of the column as bed volume increases. Journal recommendation for a gel filtration chromatography is to have length of the column more, which is less in diameter in comparison to other type of chromatography. Another factor is selection of media. Media should be porous matrix, it should be inert, physically and chemically stable. So while choosing a media, one should always consider two important factors. One is, what is the aim of the experiment? Is it group separation or purification of target molecule? What is the molecular weight of target molecule and other impurities to be separated? There are three functional components of the column: pore volume, void volume, and bed volume. So selection of media is important. Another thing is composition of the buffer. Selected buffer should not have any effect on the biological activity of target molecule, as well as it should not disturb the stability of the molecule to be separated. Since the high quality of detergent or denaturing agents in buffer can cause dissociation of subunits or cofactor of proteins and enzyme. So these kind of buffers should be avoided. Extreme range of buffer pH should also be avoided, otherwise it could disturb the conformation of the beads of the resin. pH of the buffer should always be away from the pI of the target protein. Buffer should be prepared in high quality double distilled water and it should be filtered properly so that it does not have contaminant that can choke the column. Another important factor is sample volume and concentration. Prior to loading, the sample should be filtered. It should be clear and free of debris. If sample is not clear and contain large size impurities, they can block the column. Sample may be prepared in another buffer since sample buffer is exchanged during the separation. Sample should be concentrated but not to the extent that solubility is disturbed. Highly viscous samples may result in poor separation and they may lead to peak broadening due to irregular flow pattern. Sample should not be essentially diluted because it may get diffused and no separation will be achieved. Sample loading is also an important factor. It is always better to inject sample through an auto sampler. If you are loading sample manually, one should take care that surface of resin is not disturbed. If surface is disturbed, irregular peaks will be obtained, resulting in poor separation. Flow rate is also an important factor. Elution in gel filtration is done isochronically using single buffer system. Flow rate should not be very high or very low. High flow rate will lead to poor separation because analyte will not get sufficient time to travel through the pores, while slow flow rate may lead to diffusion of sample causing band broadening. Length of tubing is also important. Length between the fraction collector and the column should be appropriate. It should be as small as possible. Larger tubing connecting the fraction collector and the column will allow mixing of different peaks on the way and it will result into poor separation. Fraction size is also an important factor. It should be small to give better resolution. If fraction size is large, say 2 ml, 3 ml, again it will result into mixing of analytes. Gel filtration chromatography can be applied for determining molecular weight of a protein or a substance, for desalting, for separation of macromolecules, and for group separation. For molecular weight determination, gel filtration chromatography is widely applied. The molecular weight of a given protein is estimated by comparing its elution volume with that of known protein standards. The standard curve is constructed by plotting log of the molecular weight of standard proteins against their respective ratios of elution volume to the column void volume, that is ratio of V and V0. This standard curve is then used to determine the molecular weight of unknown proteins. Gel filtration chromatography is also used for desalting purpose. The salt molecules and buffer components are several times smaller in comparison to macromolecules. For this reason, gel filtration chromatography using a resin with small size exclusion limit like G10 or G25 can be used for desalting and buffer exchange applications. The desalting is the process of salt removal from a protein sample, while in buffer exchange, a protein solution is replaced by more appropriate buffer. This is one of the most applied application of gel filtration chromatography. The separation limits for resins are generally in the range of up to 10 kilodalton. The resins with large exclusion limits are not suitable for buffer exchange or desalting application. The third application is separation of macromolecules. Gel filtration chromatography is commonly applied in the research lab for the separation of proteins and peptide. It is also applied for detection and separation of oligomers. Another important application is group separation. Gel filtration chromatography can be used for fractionation of crude sample into low and high molecular weight protein groups for further purification. What are the precautions one should take while performing a gel filtration chromatography? First is selection of appropriate media is very essential. It should be selected according to the molecular weight of a molecule to be separated. Suppose you want to separate a high molecular protein, say 100 kilodalton and 200 kilodalton protein, and you have chosen a media G50. G50 can have range up to 30 kilodalton. It can separate molecule up to 30 kilodalton. So in this case, this is a very wrong choice. Everything will come into void volume. Nothing will get separated in G50 if the molecular weight of a protein is very high. Similarly, if you take say G10 or G25 column, you cannot separate very even low molecular weight proteins. They are only for salt exchange or buffer exchange. So media should be selected according to the target molecule to be separated, according to the molecular weight of target molecule to be separated. Another important precautions one should take is column length. Column length should be quite huge, quite big. Length should be big. If the column length is small, say this small or this small, separation will be very poor. Why separation will be very poor? Because you have loaded the sample on the top, but the sample is passing through the column and it is not getting enough time to get separated, and it will come out without getting separated. So column length should be big in case of gel filtration chromatography. It should be large, and diameter should be small. Flow rate should also be appropriate. It should be such that it is not that slow that it will allow diffusion of the sample, or very fast resulting into the poor resolution. If it is very slow and column is large, your molecules will pass very slowly, and if they are passing very slowly, they'll get diffused inside the column only. And if you are getting diffused inside the column, you will have a huge broad peak with no separation. And if flow rate is very high, say 12 ml or 18 ml per hour or say 24 ml per hour, what will happen? You have loaded the sample will not have enough time, it will come out like this. If it is coming out like this, it means it is not passing through the pores nicely, it is coming out just under the pressure of flow rate. So again there will be a poor separation, poor resolution. Sample should not be precipitated before loading. When you are loading the sample, sample should be clear, should be filtered, but you have to choose pH and ionic strength of a buffer in such a way that it don't get precipitated. If it is precipitated, then your purpose of separating your molecule get defeated. Why? Because you want to have your protein in a natural form, in the native form. If it is precipitated, you will not get correct P. If it is precipitated, it will block your column, and again you have to pack the column again. So that is an important factor. Now students, let us summarize what we have learned in this module. We have learned what is gel filtration chromatography. It separates the macromolecules on the basis of their size, and wide range of gel filtration media are available in the market, which one can choose and then separate different molecules on the basis of those medias. Column dimensions, media, flow rate, tubing length affect the separation and resolution of gel filtration chromatography. Gel filtration chromatography is very useful. It is applied at many places like molecular weight determination, purification of proteins and peptide, desalting and buffer exchange.